Dear students, in the previous lecture I gave you a brief introduction of landfills, what they are used for, what are the typical problems in a country like India, and definitely a very sad incident that I shared about the Ghaziabad landfill slide. So today what we are going to do is we are going to talk about the leachate composition, what are the kind of microbes, what are their processes and how do they degrade the leachate, because leachate is a big trouble. And then hopefully we will get to a portion where I will be talking about different kinds of, at least 3 different research that have happened on solid waste management focusing on landfills. So let's get started. So in the last class I talked about the leachate and I mentioned that leachate is the liquid that collects in a landfill site over time. Now if we have a landfill such as this one, let's say this is a typical landfill cell. Now in this landfill cell over time we will have a certain solid portion. Now this obviously the ratio of solid to liquid to gas depends upon the composition of the waste that was put in and also upon the climate. So and then we will have a liquid portion and then we have a gaseous portion. Now the liquid portion is what is referred to as leachate. Typically leachate consists of if the leachate is old, depending on its age, it has organic materials in it which are very hard to degrade. So it might have heavy metals in it depending upon the kind of mechanism happening in the landfill, its health and its age, and it might have other compounds in it. Most of them are not good for health, whether it is our heavy very complex organic material, whether it's the heavy metals or it's the other compounds. Leachate degradation is still a hot topic of research. So let's get started about leachate. Now composition of leachate depends on the climatic condition, depends on rain, moisture and temperature. So for example, after we have sealed the landfill, ideally the liner here is completely impermeable, which means that its permeability of liner is 0. And when we seal the landfill from the top, we add other liners and we typically add mud over it. Now its permeability is also 0. So typically, ideally this is a complete batch reactor, perfect batch reactor, completely sealed. However, there is always some rain that percolates into over time, or maybe if there is some flooding and then the groundwater can also percolate in from imperfect liner, and when this happens the liquid content increases. So the rainfall that percolates in or the groundwater that percolates from side depending on changes in the groundwater flow, then this liquid acts as like a washout liquid, and it as it percolates from top to bottom, it dissolves whatever can be dissolved, heavy metals, organics, humic acids, whatever can be dissolved, it dissolves and then it pools here at the bottom or over at different heights as leachate. Now this leachate builds up over time, obviously as every season the rainfall is new, and as the biodegradation of the waste also continues, we have more water that is generated and thus more leachate is generated. Thus most landfills will have a proper leachate removal system. Typical leachate removal system would include pipes from which the leachate is pumped out regularly. So basically the formation of leachate and the composition of leachate depends upon the rainfall, the moisture, the temperature, because the temperature governs the microbial activity, also the degradation of waste, whether it's anaerobic or aerobic. If it is anaerobic, then the water is consumed, and if it is aerobic, then water is released. Obviously if it is aerobic, then oxygen will take electrons from hydrogen. Alrighty. Now this is your once the leachate has been pumped out from the landfill. Now these are the landfills and leachate has been pumped out. They typically end up in a leachate collection system, and this is a picture. This is not India, but this is your leachate collection pond. Then this leachate is taken for further treatment. Further treatment would include evaporation of the water content. So we want to reduce the volume of the leachate. It might include different kinds of purification process to get rid of heavy metals, to get rid of other contaminants, and then eventually either we might evaporate the leachate or we might incinerate the remaining components. Okay. I have mentioned this, that the composition of waste depends upon the temperature, the climate, and the rainfall. Definitely it also depends upon the kind of degradation. So pretty quickly, I hope you gather that the degradation happens over time, and since degradation happens over time, at different time, we have talked about it in the previous lecture, we talked about 5 phases of degradation of waste. Now these 5 phases progress over time, so depending on the age of the waste, the kind of leachate produced would change. For example, when we have the aerobic, the first phase of degradation, then we will have more water produced, and thus leachate will be rich, will be low in concentration of recalcitrant compounds, and there will be plenty of water, but over time leachate will get more and more concentrated. Then it also depends on design and operation of landfill. Depending on the design of landfill, the amount of waste it receives and is filled into it, the way it is operated will also determine the concentration and the composition of leachate. And then climatic condition we have all talked about. So and now speaking of waste age, as the waste ages, as the waste undergoes different kinds of degradation, we will notice that different kinds of contaminants are found in the leachate. In the first phase, where we have aerobic, we will have little of all, little of COD, BOD, ammonia, chloride, and heavy metals. But as it enters the acidic phase, where the pH drops, we will see that we will have lot of heavy metals. So the heavy metals will start increasing. This is only the time when heavy metals peak, and we will also see that the COD and the BOD will increase, because now the waste has been created to a level that has been packed to a level, enough sufficient level that the COD and BOD are increasing. We will also see that the ammonia would increase, the chloride, the salts would increase. So now this is, because this is an anaerobic process, one of the major reason why everything is increasing in concentration is because the amount of water that was present in leachate in the landfill earlier is now being consumed to carry out the anaerobic acidic degradation. And because the pH falls in this acidic degradation, we notice that the heavy metals get dissolved at a higher level. Now, when it enters the third phase, which is the initial methanogenic phases, this is where the BOD undergoes a rapid drop. So this is, ever since then the BOD remains minimal. Also the COD starts dropping. So this is where the real BOD COD of leachate start dropping. Another thing to note is that the salinity undergoes only a slight drop, and even our ammonia undergoes a slight drop. The metals have reduced because now the pH is coming back to nearly 7. Now here on the stable methanogenic phase on, the pH might actually go above 7. So definitely we are not expecting heavy metals. Also interestingly, the BOD has reduced completely, the COD has reduced considerably. It is in these phases, from the phase 3rd to phase 4, that we see considerable decline in the solid content. Now similarly, in the 5th, 6th and 7th phase, there will be a continual decrease, and in the 8th phase, when there is re-infiltration of air, then we notice that finally our ammonia gets oxidized and we have perfectly stable landfill. Now why is there a re-infiltration of air? This is very important to oxidize ammonia and other reduced form of compounds that are product of all other degradation and are awaiting the degradation. Now this reaching this stable phase might take, again as mentioned earlier, geological scale of time, and we might not see it in our lifetime, but this is what we expect. Okay, so what does leachate constitute of? Definitely there is water. That two things, water, because it's liquid, there's definitely water, and the water comes from two sources. One is the waste itself and the other is rainfall. If we remove the water, we dehydrate it, then we are left with some cellulose, hemicellulose, protein and lignin. Please note that cellulose, hemicellulose and lignin are very recalcitrant. Lignin particularly is not amenable for degradation, takes forever long to degrade. Most of it is cellulose, which undergoes cellulose, anaerobic cellulose degradation, and we'll talk briefly about it. And then there is hemicellulose, which differs from cellulose in its structure and morphology. Now each of these components, the dry components, will be eventually converted into methane. So let's look at the total methane potential. It's highest in cellulose, followed by hemicellulose, protein and lignin. Okay, now what are the different kinds of degradation pathways that microbes can take when they are degrading waste in the landfill? They can undergo the aerobic degradation, they can undergo fermentation, they can undergo anaerobic respiration. We have briefly talked about, we have quite talked about these processes in earlier lecture. So if you are not clear about either aerobic degradation, fermentation or anaerobic respiration, please go ahead and review some of the early lectures. In aerobic degradation, obviously oxygen is your electron acceptor, and we have very high generation of energy. Depending on the cycle, it will undergo glycolysis, it will undergo citric acid cycle, respiratory chain, and there is no scope of methanogenesis. In fermentation, there is no other electron acceptor available. In methanogenesis, we have carbon dioxide that serves as electron acceptor, but in fermentation we have no electron acceptor available. So everything is getting reduced. We have no scope of citric acid cycle and respiratory cycle or methanogenesis, and we have fermentation happening, and the total energy output from air compared to aerobic falls to nearly 1/3, less than 1/3. Then anaerobic respiration, carbon dioxide serves as electron acceptor, it gets converted into methane, and thus this process is also called as methanogenesis, and we have methanogenesis undergoing, and depending upon how much CO2 is there, how much acetate is present, we will generate some energy. Okay, we have briefly talked about this before, but let's go through this again. The landfill undergoes 4 distinct phases, and then there is a 5th phase of stabilization, microbial processes. So the first microbial processes is hydrolysis. So remember, if you remember, nearly 51% of waste is in leachate is cellulose, 11% is hemicellulose, and lignin is 15.2. All these 3 are very long, very complex polymers. So nearly 96% of the leachate dry weight is complex polymer. So in the first phase of degradation, microbial degradation of leachate waste, what we have is the high molecular substances, the polymers, really complex polymers, which are nearly 96% of the dry weight, they are degraded into low molecular compound. For example, the cellulose consists of monomers of cellobiose. So these cellobiose units, cellulose are degraded into cellobiose unit, which can be very quickly degraded. Now in the second phase we have acidification. So now the daughter products, these low molecular compounds, undergo acidification, and in acidification, organic acids are formed, like propionic acid, butyric acid, lactic acid, acetic acid, along with some ethanol, hydrogen and carbon dioxide. So in this phase we generate acids from these low molecular compounds. First we break the long chains, and then we acidify them. Microbes convert them into, oxidize them into acids. Some carbon dioxide and hydrogen will also be produced. In the third phase we have acetogenesis. This is where acetic acid is being produced. We will give you a brief example very soon. And so we have hydrogen, carbon dioxide and acetic acid. In the fourth phase we have methanogenesis. So all these oxidized forms of carbon, now they serve as electron acceptor. Eventually we are left with methane and carbon dioxide. So this is your typical flow chart for degradation of leachate. You have organic substances such as carbohydrates, lipids and protein, and in case of leachate you will have cellulose, you have hemicellulose and lignin, which together form nearly 96% of leachate dry weight. They undergo hydrolysis, they make smaller compounds, sugars, fatty acid, amino acids. They undergo acidification, they will form acetate, hydrogen and carbon dioxide, or they will form organic acids. The organic acids undergo acetogenesis. So all these organic acids are converted into acetic acid. Now this acetic acid undergoes methanogenesis and forms carbon dioxide and methane. This is degradation of the dry weight of your leachate. Now let's look at how microbes do the cellulose degradation. Here is a very nice picture of Clostridia thermocellum. So this here, what you are seeing is actually a cellulosic microfiber. and these little tiny things here are your bacteria. So the bacteria are eating your, this is one bacteria, one cylindrical rod is one bacteria. These bacteria have clustered around, cornered around the cellulosic microfiber, and they are degrading it. Now this is another picture of another microbe, F. succinogenes, which are these small fibers, microbes. They are sticking to cellulosic fibers and they are degrading it. So we have very good evidence showing how microbiologically cellulose is degraded, and remember 51% of leachate dry weight is cellulose. Now this is very important to know that cellulose in aerobic, under aerobic condition is very recalcitrant. So unless it is oxidized due to fire, so unless there is a fire, cellulose will not be oxidized. It is very recalcitrant, but then over under anaerobic conditions it undergoes as hydrolysis followed by acidification followed by acetogenesis, methanogenesis, and is degraded. Okay. Now this is from a very cool study that was done recently in Europe, and what they did was they looked at pH, water content, methane production at 1 meter depth, 3 meter depth, 7 meter depth, 11.5 meter depth and 17.5 meter depth of a landfill. So they noticed that as we went below in the from 1 meter to 3 meter, the pH dropped, the water content increased because now the water is going from top to bottom, and the methane production activity sort of decreased. Oh well, methane production activity, we can say that the hydrogen to carbon dioxide ratio decreased from 2 to less than 1, much less than 1, and the number of the concentration of acetate decreased slightly. Now this is same study, different table. We have at deeper depths 7 meter, 11.5 meter, 17.5 meter. We notice that the organic matter content drops from 7 to 11.5 meter. So here we have a lot of organic matter that can degrade, but it has, the one at 11.5 meter is more aged. So with age the waste has been degraded and the BOD, the COD have dropped, and then after time they remain constant. So if you go back to your diagram here, if you go back to your diagram here, after a while the BOD and the COD, they become constant. So now organic matter has become constant, the moisture content dropped. So initially it would have increased and dropped, and then it slightly increased because now it is collected at the bottom. Moisture content at any time depends upon the weather and the flow of water around the landfill. Monosaccharide content at the nearly top, 7 meter is not top, but this is newer waste than this one and this one. So monosaccharide content in the first two is higher, and then here by the time it is really old it has degraded. The Clostridia, now Clostridia is one of the microbe that was first identified to degrade cellulose. Members of Clostridia were identified to degrade cellulose in a single culture. So this is your Clostridium thermocellum. So they noticed that Clostridia increases with age from 7 to 11.5 meter, and then when all the organic substances have been hydrolyzed and much what is left is just methanogenesis of the acetate that has been formed, then they drop. Okay, so we can briefly summarize our degradation of leachate and waste in the landfill as the complex polymers. They undergo hydrolysis and they are broken into smaller sugars. The sugars are then fermented into acid. So this is acidic, the pH drops here rapidly, and the next step is when these acids are now converted into acetate. So this is acetogenesis phase. This is very important phase because after this acetate is converted into methane gas. So remember the Bangalore landfill, there was a lot of BOD, and thus there was very good concentration of methane that was produced. And this is sad because methane is very potent greenhouse gas, and we definitely do not want our people inhaling a lot of it. Also it is highly inflammable, so there is a high chances of accidents and fires. Okay, now let us look at microbes that were responsible for degradation. With different temperatures, we notice that different kinds of communities are formed. So as the waste age, the temperatures change, and as the temperature in the region changes, the temperatures change. So Clostridium, the cellulose degrading microbes are, well, not all Clostridium are cellulose degrading, but the Clostridium genus can be found in psychrophilic to thermophilic. So from really cold temperatures to really hot temperatures. The methanogens are also very diverse, and from very cold to very hot temperatures they exist. Then there are Trichoderma, Aspergillus, Curvularia and Fusarium that like thermophilic conditions, really hot. Fibrobacter like 30 degrees Celsius, Proteobacteria likes mesophilic temperatures. Now the moisture content, methanogenesis like when there is 500 to 600 gram of moisture per kilogram of waste. Depending on pH value, methanogens prefer that the pH should be around 7. So when there is anaerobic acidic degradation, methanogenesis will not flourish. They will actually wait for pH to stabilize around 7 before they flourish, thrive. They might survive under extreme conditions. Now again, Trichoderma, Aspergillus and Curvularia and Fusarium, they like slightly alkaline conditions. So you can imagine that they will come over after methanogenesis has been started. And then we have Fibrobacter and Proteobacteria which like around 6 and around 7. Okay. Now with sample age, we have old refuse samples and young refuse samples. So this is your old high water age, higher depth. This is lower water age, lower depth, or old. This is your old landfill site, landfill cell. This is young landfill site, young landfill cell. In the old, you have lower concentration of acetate because most of it has been converted into methane. You have lower microbial activity because microbes have already done their job and now they are not very happy because there is very little food left. You have a lower level of cellulitic bacteria because whatever hydrolysis was required to happen already happened a long time ago. In this kind of case, you will notice that you will have lot of bacilli, lot of bacillus, bacillacea, and you will notice extremophiles. One thing I did not mention in the previous one of the previous slides is that at 17.5 meter depth, the researchers of this study notice extreme halophilic condition and very high temperatures. So this very high salinity and very high temperature. So as waste ages, or in other words, if we are doing vertical filling of trenches, then with depth we notice that the temperature increases and we have extreme conditions. So we have extremely salty, extremely alkaline conditions. And then again, lot of bacillus like DNA sequences. These are bacillus like organisms. We do not know what they are because they have not been cultured. We notice lot of proteobacteria, we notice methanocyta, hydrogenotrophic, hydrogen forming and formate using methanogens. So in the young refuse samples, on the other hand, because they are younger samples, they will have lot of organics to degrade and lot of cellulolytic bacteria to carry on the hydrolysis, and once the hydrolysis has happened, the sugars have been converted into acids and acetate into acid into acetate, then that is why we will have lot of acetate, and then overall we will have a very high amount of microbial activity. Such conditions are not favorable for proteobacteria which like the lower microbial activity conditions of old refuse samples. These conditions are very favorable for methanosarcina because of higher concentration of acetate. They are also favorable for acetoclastic, hydrogenotrophic and formate using methanogens. Now a note that they have placed here, it is pretty nice note, let us look into it. Significant structure changes of the methanogenic community happen shortly after waste burial. Now why would this happen? Because right after burning the waste, we have cut off the supply of oxygen, and now the degradation has started and methanogenic community will start shifting. Now let us look at depth. Now here depth is representative of the age of the waste material. So we can notice that in upper waste, we will in the upper waste layers we will have a condition similar to young refuse sample. In deeper waste layer we will have conditions similar to old refuse samples. So the deeper layers are the older ones, they have higher waste age. The upper ones are the newer ones, they have lower waste age. So in the deeper ones where the acetate has been consumed, the microbial activity is low, cellulitic bacteria is low, methanocolons would survive. In upper waste layer where the samples are young, microbial activity is high, there is lot of acetate, methanosarcina would survive. Now high pH. Now why would a landfill have high pH? Remember, one of the very important phase of degradation in landfill is the second phase where we have lot of acids producing. So pH should decrease, but why does pH increase? pH increase would happen when we take our solid waste, we incinerate them, so we have ash from there. Now when that ash is put into a landfill, that ash might increase the pH. In this case, the microbial community will be dominated by bacillus. Now let us look at the organic matter concentration. When we have high organic matter concentration, obviously we will have high diversity of microbial communities. Now let us look at leachate. If we are doing full scale recirculation of leachate versus if we have a closed landfill. If we have a closed landfill, the conditions would be relatively stable. These conditions are very nice for methanocyata, but not for methanosarcina, which loves a lot of organic material, lot of acetate. Again, because the conditions are stable, the proteobacteria would survive, thrive. In full scale recirculation of leachate, what happens is that with the leachate recirculation, the organic matter increases. So basically we have leachate that is being collected down and then it is being recirculated. So every time it comes down, it collects lot of organic matter and it accumulates lot of acetate. These conditions are very favorable for methanosarcina, which loves high acetate conditions, and these are not favorable for proteobacteria, which likes stable low microbial activity conditions. Now let us look at temperature. At high temperature from 40 to 50 degrees Celsius, we will have low archaeal diversity. At relatively lower temperatures compared to this, we will have high microbial diversity. Now contaminated sites. Some of the sites, some of the waste are very bad and we call them as contaminated sites. In a landfill, if we have, for example, an industrial waste landfill or hazardous waste landfill, then these would be a contaminated landfill. So if there are hydrocarbons, PAHs, PCBs or heavy metals or other hazardous waste, proteobacteria will thrive because overall microbial activity would be low. Depending on the contamination intensity, we will range from lowest microbial diversity and lowest enzymatic activities in areas with highest pollutant concentration and vice versa. Now let us look at microbial degradation. Who is doing the degradation? Typically we will notice lot of Clostridia, so a lot of hydrolyses. We will notice Fibrobacter, Proteobacter when the microbial activity is low. Methanogenic archaea would be dominated by Methanomicrobialis and Methanosarcinalis, so which is lot of methanogenesis happening. In the next lecture I will take over and talk to you about 3 studies on the microbiology of landfill sites and what are the insights that people have recently generated from the microbiology of landfill degradation. That's all for today. Thank you very much.